Thanks very much, Dan, and also your co-organizers for having me here and talk to you today about a modification which I think is maybe not as well known as some of the other post-translational modifications. So I'll take you through several oxidative protein modifications today and what their role is and how they're controlled, particularly in plant immunity. Now, in the cell, particularly the plant cell, there are many sources of the production of reactive oxygen and reactive nitrogen species, or ROS and RNS, as I'll call them. The cell wall and the apoplast produce a lot of ROS and RNS. Of course, the chloroplast, where photosynthesis takes place, is a hothouse for the production of these species as well. The respiratory chain in mitochondria is a strong producer of ROS. The peroxisomes, a lot of metabolic processes contribute to the production of ROS and RNS. And even the ER and the cytoplasm are sources of accumulation of these different reactive species. Now, particularly stresses, biotic and abiotic stresses, induce the accumulation of these different ROS and RNSs. And this can be very excessive, as shown in the picture over here. This is an example of plants infected with Pseudomonas undergoing a cell death response. So you can see associated with that, you get strong accumulation of reactive oxygen species, in this particular case, hydrogen peroxide, as well as nitric oxide accumulation. So those are some of the species that you may encounter under such a stress. Now, when we look at redox signaling as a whole in the cell, we can sort of schematicize it like this. The accumulation of these species usually has an interaction with stress hormones. Often it facilitates their production, and vice versa. These hormones facilitate the production of more reactive species. And that all interacts with cellular redox couples within the cell, particularly glutathione and ascorbate. Both are fueled by an underlying NADPH dependent system. And this essentially can buffer some of those different reactive species within the cell. But both of these, both the ROS RNS and the redox couples, have a particular output that can be sensed by different proteins, which I will call redox sensors in my talk here. And redox sensors contain sensitive cysteine residues that can either be in an oxidized state or in a reduced state, and that can reversibly switch between these different states. Now, there isn't a single state for a cysteine residue. There's actually multiple states that can be attained by this quite complicated single amino acid. And so we can start with the free thiol, the completely reduced state, and work our way up an oxidative ladder in which you reach total oxidation over here. And so we go through phases of, for instance, the addition of nitric oxide, which we call S-nitrosylation. I'll show you a little bit of this later in my talk. A higher oxidation state is S-sulfonation, the addition of hydroxyl group. You're most familiar, I'm sure, with the disulfide bridge, which sits somewhere over here in the ladder, together with S-glutathionylation, where you add a glutathione directly to the cysteine residue, which essentially also a disulfide bond. And then you can go even higher to S-sulfonated states and S-sulfonation. And S-sulfonation is important to mention that that's a state of oxidation that can no longer be reversed. And so it's usually associated with pathologies in organisms. Whereas all of these other states can be reversed. Now, the reversal is often performed by a family of thioredoxins. It's a superfamily containing many, many different enzymes, particularly in plants. And thioredoxins contain this typical fold shown here in the crystal structure of thioredoxin 1 that contains an active site that contains two cysteine residues. They're over here in the crystal structure. And those two cysteines sit at the end of an alpha helix, as you can see here, and that creates a dipole moment. It also creates a hydrophobic area, which drives down the pKa of these cysteines, which makes them very nucleophilic, so very reactive. They're also solvent exposed, so they're ready to interact with oxidized cysteine residues that they may target. And so their reduction of oxidized cysteines will look something like this. So a reduced thioredoxin comes in and essentially transfers its reducing power to this oxidized cysteine in the target protein, which then becomes reduced. The thioredoxin instead becomes oxidized and can be recycled with the power of NADPH. And so there's this dedicated arsenal of thioredoxins that can reduce these different modifications that I've just introduced you to. So sulfur-redoxin is really the only enzyme known to be able to reduce the S-sulfonated state back to the sulfonated state. Glutaredoxin, well known to take S-glutathione-related residues and reduce them down to the free thiol. And the Thioredoxin family tackles probably the largest amount of modifications. It takes all these different oxidative modifications and reduces them to the free thiol. Now, of course, the Thioredoxin family, I think in this case, because they reverse these modifications, they now allow these oxidative modifications to become signaling tools, because you can now create an on-off switch. And so what we ask in my lab is, well, do these thioredoxins do more than that? Do they just provide reversibility, or is there more to them? And so I want to highlight two examples to you today, two brief examples of other roles they may play. So the first one that I'll highlight is a role of thioredoxin in reducing S-nitrosylated proteins, so proteins in which cysteines are modified by nitric oxide. And so to do that, we took two different mutant backgrounds, one called GSNO reductase 1. This is a mutation that causes accumulation of a natural nitric oxide donor called S-nitrosoglutathione. Basically, that's just S-nitrosylated glutathione. It's a very good NO donor, so it passes on NO onto protein, target proteins, and forms so-called protein SNO, or S-nitrosylated proteins. And so these mutants have highly elevated levels of these protein SNO. We also use NOX1 mutants, and NOX1 mutants have a mutation that causes elevation of L-arginine, and L-arginine is a precursor of nitric oxide, and so consequently, these plants have very high levels of free nitric oxide. That also functions as a donor for S-nitrosylation on proteins, and so both these plants have elevated protein SNO levels, and both of these plants, because of that, are immune compromised. And so we looked at the role of thioredoxin H5 in these plants, because thioredoxin H5 is a immune-specific thioredoxin in Arabidopsis. And you can see that just over here. The expression of thioredoxin is off in the resting state, but after activation of immunity, here by adding salicylic acid, which is an immune hormone, you can see that its activation, its gene expression is activated quite strongly. However, you can see in the GSNO reductase mutant and the NOX1 mutant, where they have these high levels of protein SNO, activation fails. So what we did is we brought back in, under a 35S promoter, we brought back in thioredoxin H5 in these two different backgrounds and observed what would happen. And what we found is something that was quite surprising. Knowing that thioredoxin H5 can reduce S-nitrosylated proteins, we thought we would rescue both of these backgrounds. And so we did the test by infecting these plants with a pathogen. And so you're looking at pathogen growth here of Pseudomonas. Here's growth on wild-type plants of Pseudomonas. It does reasonably okay. But on GSNO reductase mutants, that are immune compromised, you see much higher growth. Note this is a log scale, so 1.5 log more growth here. But when we put in thioredoxin H5, which is supposed to reduce all those S-nitrosylated proteins, we had no effect. However, if we did this experiment in the NOX1 mutant background, which are also immune compromised, we could rescue that level of immunity back to wild-type levels just by expressing Thioredoxin H5, and that was associated with a reduction in the amount of S-nitrosothiols proteins within the cells. It was associated with a full rescue of immune signaling, and as you can see here, by immunity. And so, making a long story short there, what we managed to show is that in addition to providing reversibility, actually these thioredoxins probably provide a lot of specificity to signaling, because they don't target just any oxidized protein, and in this case, not just any S-nitrosylated proteins. They target specific classes of S-nitrosylated proteins. So what we think is that Thioredoxin H5 only targets proteins which have been modified by nitric oxide, a free nitric oxide, whereas those that are modified by the natural donor GSNO are not targets of thioredoxin. And instead, those are probably regulated by GSNO reductase itself, which indirectly controls the levels of these proteins by governing the levels of GSNO. Okay, so in addition to reversibility, we think they provide specificity. So I'll give you another example where we find another role for a thioredoxin. So the thioredoxin I've just discussed with you is a fairly simple thioredoxin with a single active site. But there are also redoxins within the genome that encode for more complex molecules, such as this one. This is nucleoredoxin 1. Nucleoredoxin 1 have 3 active sites, 2 of which are probably the real active sites because they contain the cysteine residues there and there, whereas the thioredoxin-like fold misses the second cysteine. We explored what these different molecules do. We tested the activity. The activity of this protein is to reduce oxidative modifications, as we would expect. It's widely localized throughout the cell. And we found that, like Thioredoxin H5, it can be induced following immunity. So here's infection by a pathogen. You see that within 24 hours, there's very strong induction of this gene, which we can't find, of course, in the knockout mutant. And this is also seen at the level of activity rather than gene expression. So if you look at the protein itself, you could see that addition of pathogen or pathogen infection, you suddenly start to see substrates of NRX1 accumulating, whereas in untreated cells you see very few substrates accumulating. And this assay is performed essentially by capturing those substrates when they're interacting together with NRX1. And so this suggests it has a role to play in plant immunity. And indeed, when we look at NRX1 mutants, 2 different alleles here, we see that they have constitutive immune gene expression, which we can rescue by adding back in NRX1 gene. Associated with that constitutive immune gene expression, we see also constitutive immunity. So again, here you're looking at pathogen growth, Pseudomonas growth on wild-type plants, and plants that have been induced for immunity. So you see that the immune response has been launched. But an NRX1 mutant, already quite resistant, and immunity cannot be induced any further. And that's all dependent on salicylic acid signaling in this immune response. So we were very curious to know what the substrates are for NRX1 to maybe uncover how it established these immune phenotypes. So to do that, we followed the following purification strategy. And so I've just very deduced the NRX1 protein to a simple diagram here of its active site, or one of its active sites. So this is what it would look like. The active site has a cysteine, which is in a sulfhydrated state, so very nucleophilic, and that faces the target protein. Interaction with that target protein reduces the oxidation in that target and establishes a link between the nucleoredoxin and its target. And I want you to note that this is a covalent link between these 2 molecules. And the second cysteine then resolves that link, causing the NRX to become oxidized and the target reduced. So what we did is we mutated this second cysteine residue in all the active sites. And so that would be the molecule that you see here on the right. And in this molecule, of course, the first cysteine can still make the attack. But since the second cysteine is lacking, all the targets get covalently trapped onto the thioredoxin or onto the nucleoredoxin. And so consequently, if you measure the disulfide reduction activity of these different molecules, you can see that the wild type has strong activity, whereas a mutant version has very low activity because all the substrate gets stuck onto it. And so we purified these substrates, submitted them to mass spec sequencing, and came up with the following list. We found that highly overrepresented amongst our targets were proteins that are involved in the electron transport and energy pathways. So this is, of course, very promising because these pathways are highly associated with reactive oxygen species. And further down the list, we also found responses to stress and abiotic stimuli, which was expected because our samples were derived from plants that were pathogen infected. And when we looked further into that list of targets, we fished out essentially a whole pathway. Although not all of them were completely significant hits, they kept showing up in our analyses, in our repeats. And so what we really got out of that analysis was essentially the whole hydrogen peroxide detoxification system, which consists of several enzymes. So hydrogen peroxide in the cell is normally detoxified by catalase, which does that independently, but also by ascorbate peroxidase with the help of with a reducing power of ascorbate. And that functions in a cycle with monodehydroascorbate reductase and dehydroascorbate reductase, which essentially provide the ascorbate for ascorbate peroxidase to use. And so we found 3 different catalases as targets of NRX1. We found APX1, we found MDAR, and DHAR2 and 3 in our analysis. And so just for the purposes of time, I'll focus on catalase here. So we want to see whether catalase is indeed an oxidized substrate. So what we did is we tried to analyze the oxidation state of catalase. To do that, we start out with an extract with catalase protein in it, and that will have, of course, proteins with residues that are free thiols or residues that are oxidized. If you unfold that protein, then block the free thiols, you only have the oxidized thiols still into play. You can reduce those just by adding DTT, so you have free thiols again, and then you can label those with biotin and detect the biotinylation. So that's what we did, and it's what you see down here. Just to start with maybe untreated plants, we saw that in wild-type plants you have a little bit of oxidation of catalase compared to the total amount of catalase, whereas in NRX1 mutants we see slightly more oxidation. If we don't add in DTT, so we don't switch here from the oxidized to the reduced state, so we won't be able to label with biotin, we don't see anything. So this is key because that shows the specificity of this assay. But what you can see is then that infection with a pathogen or treatment with methylvilogen, which is an oxidative stressor, increases the amount of oxidation that you see, but particularly in this NRX1 mutant, suggesting that indeed catalase is oxidatively modified, and this may be held down by the action of nucleoredoxin. So we're quite surprised by this because, of course, catalase is part of a very oxidative environment as it detoxifies hydrogen peroxide. So we're surprised that it was also modified, and it may actually do something to its activity if nucleoredoxin needs to reduce it. So we looked at leaf catalase activity in our different mutants. And compared to wild-type plants, indeed, nucleoredoxin mutants, where we have more oxidized catalase, have much lower catalase activity. And here's just a control of CAT2-3 mutants, where catalase activity is almost completely knocked out. And if we add back into these extracts, if we add back into that recombinant protein, either mutant NRX1 protein or wild-type NRX1 protein, we get the following effects. So you add wild-type recombinant NRX1 into a wild-type extract, we improve catalase activity, and strikingly, in an NRX1 mutant where the catalase activity is knocked down, we can rescue activity back to wild-type levels just by adding in recombinant nucleoredoxin, whereas we can't do that in a catalase mutant. So these data suggest that NRX1 regulates oxidative stress responses through the action of catalase. Indeed, we see in NRX1 similar phenotypes as catalase itself. It's very sensitive to oxidative stressors, such as methylviologen. You see more cell death occurring in response to oxidative stressors than wild type, and so that looks very much like a catalase mutant phenotype. And so we suggest that the following is going on, that proteins that are normally, hydrogen peroxide is normally detoxified by catalase. In that process, catalase gets oxidized, and nucleoredoxin is necessary to bring catalase back in a reduced state to keep it in an optimally functional state. So just to summarize what I've told you, so I've shown you that thioredoxins introduce reversibility, but in addition to that can create specificity and selectivity in signaling, and they provide, surprisingly, protection for antioxidant system. And I'll show you that by introducing nucleoredoxin 1 as a reducer of catalase activity. And so I should really thank for all the work that I showed you today, my former PhD student and now postdoc Sophie Nisha, as well as my former student Romana Kayani and a visiting student Sylvain Guillinot. Thank you very much for your attention.